It's a pleasure to be here. I came early, so I spent a few days in Scotland, which is something I've wanted to do my whole life. I got to play golf at St. Andrews. You're looking at a very happy man. Okay. When I start, we've been working, one of my very first experiments when I started in graduate school was on the effect of mineral nutrition and health. So I've been doing this a very long time. We have a good group at Ohio State that works in mastitis. And so again, we've been doing this a long time. And whenever I give this talk, or talk like this, I need to remind people, nutrition does not cause mastitis. A bacteria infection of the mammary gland causes mastitis. Nutrition, bad nutrition, allows that infection to get going and develop into mastitis. But you can feed the cow perfectly. If you still do everything wrong, dirty cows, poor milking hygiene, all that, you're still going to get mastitis. So nutrition is just part of the mastitis control system. And I'm going to talk about two things. One is the effect of nutrition to optimize immune cell function. In other words, what nutrients do we need to feed to get immune cells to work well? And when we talk about immunity, the right definition is actually host defense. It's the ability of the cow or you to fight off an infection. The second half of this, and I won't go into this too much because there'll be other talks during this conference on this, but good nutrition also reduces these things we call immunosuppressors, things that detect or detract from immune function. For example, ketones. Ketones suppress immune function. So if you prevent ketosis, you've reduced an immune suppressor and helps host defense. I'll spend more time on this, but I will touch on this as well. Okay. One thing you probably heard a lot about, if you watch TV, you hear about it for us, if you've gone to other talks, and that's this thing called oxidative stress. Every person in this room at this second is producing free radicals. I guarantee you are producing free radicals, unless you're dead. Then you're done. ROS are called reactive oxygen species free radicals. You produce those for lots of reasons I'll get to. And these are absolutely essential for life. If you stop producing them, like I said, it's fatal. Too much of these is also bad. So oxidative stress doesn't mean there's none of these. It means it's in the right balance. And we get the right balance by feeding adequate antioxidants. So again, our goal is not to eliminate these free radicals. It's to keep them in balance for the given situation. I'm not going to go into this a whole lot. This is, when you breathe, you need oxygen, you consume oxygen. This is oxygen metabolism. And during normal oxidative metabolism, sometimes you get this compound called superoxide. And that's that oxygen with a dot. That's free radical, and that's superoxide. And a little bit of that escapes. About 1% of the oxygen you consume, or I should say that rats, that's where the study's been done, about 1% of the oxygen rats consume escape as superoxide. And that's a bad chemical, has to be controlled. So this is just basal, everyday production of free radicals. The one that we get more important, I should mention, this is totally proportional to oxygen consumption. So a high-producing cow consumes a lot of oxygen. A high-producing cow produces more superoxide than a low-producing cow, which may mean you need more antioxidants. The one we're more concerned with is called activated reactive oxygen species synthesis, the purposeful synthesis of free radicals. This is a very, very simplistic immune cell function diagram. It's much more complicated than this. But here's the bloodstream. This is a neutrophil. That's a primary immune cell. This is the mammary gland in this example. These little yellow guys are bacteria. So for somehow the bacteria got into the mammary gland, the first thing that happens, you hope, is signals come out and say to the blood and tells these neutrophils, come in, and kill these bacteria. This is somatic cell count. When you get infected, you want a high cell count. So these cells come in and kill. So this is called migration. It gets into the mammary gland. The next step is this PMN has to engulf or phagocytize the bacteria. You can't see it real well here, but this little yellow dot now is inside the neutrophil. It's still alive. This bacteria can multiply so much that it eventually would kill the neutrophil. So it can't stop here. The last step is what's called the oxidative burst. And these neutrophils consume massive amounts of oxygen, and they purposely produce superoxide. And then superoxide is converted to even more toxic chemicals, and that kills the bacteria. But the thing is, with this happening, superoxide concentrations increase by 1,000,000-fold. It's huge, but it has to happen, or this bacteria won't get killed. But then you have to keep it in control. And what happens is this cell eventually dies, releases all this superoxide, you get inflammation and get more severe disease. So we want this, but we want to control it. So that brings us to antioxidants. This is from the US, and the reason I don't know your soil. So the order of importance depends on soil and feeding management. Basically, any place in the world we're selenium deficient. That's almost guaranteed. So selenium is probably the most important antioxidant because you're usually going to be deficient unless you supplement it. Vitamin E, in the US, we're a hay silage-based system. Vitamin E is usually deficient if you don't supplement it. If you're a pasture-based system, vitamin E moves way down the list because pasture has so much vitamin E. Copper depends totally on soil. I don't know your soil condition, so I'm not going to say where that goes. Where we're at, soil is deficient in copper. Our feeds are deficient in copper. Your feeds may or may not be. Beta-carotene, I stuck in the middle because the data is a little bit iffy on this. It's highly correlated to vitamin E, so if the feed is high in vitamin E, it's going to be high in beta-carotene. If it's low in vitamin E, it's going to be low in beta-carotene. And this one is probably important, but it's hard to show the effect. And it might be because the basal diets aren't low enough. So it's important, but whether it's important enough to supplement, that's still debatable. Zinc depends on soil. US soils tend not to be that low in zinc, so it's a little lower down. And then these other ones, manganese, iron, these are rarely deficient enough to cause antioxidant problems. Vitamin C here is at the bottom. A healthy cow makes vitamin C. A sick cow may not. So vitamin C might move up with a sick cow, but usually it's not needed. Okay. This is, and people usually laugh when I say this is a very simplistic diagram of antioxidant systems. And it is very, very simplified. If I put the whole thing on, you wouldn't be able to see arrows. There'd be so many. The point I want to make with this is that the antioxidant system is a system. It's all connected. And you can't just think one nutrient or another. You got to think of all the nutrients. The other thing you have to remember is cells are divided into water phase, the cytosol, and the membrane, fat soluble. Antioxidants are either fat soluble or water soluble. And you can have a ton of water-soluble antioxidants, not have enough fat-soluble ones, you're going to have problems. And what I want to just point out, I'm not going through all this, but this connectivity. Copper and zinc are needed to make this enzyme called SOD, which takes superoxide, converts it to hydrogen peroxide. Selenium is in this enzyme here, glutathione peroxidase, which takes hydrogen peroxide and makes water. So if you're deficient in copper and adequate in selenium, you're still going to have lots of problems because you're going to have a buildup of superoxide, which selenium has no effect on. So again, don't just think I can feed selenium and fix everything, or I can feed vitamin E and fix everything. Everything has to be right. The other thing I need to remind you, and I don't know if I have a slide, I may have forgotten this, is antioxidants by their nature are very reactive compounds, which makes them pro-oxidants. Any antioxidant in excess becomes a pro-oxidant. So copper in the right amount helps suppress this. Excess copper actually increases radicals. So keep that in mind. Enough is enough, and too much is too much. Okay. And then vitamin E is the primary lipid-soluble, fat-soluble one. So it's important we have vitamin E down here. The metals, copper, selenium, zinc, vitamin C are the water-soluble ones. So, and you got to have both. Okay. Now, the way we think, or the way we've thought for a long time, is antioxidant nutrients control free radicals, reactive oxygen species. Keeping these in the right amount protect immune cells so they can do their job, and they live longer and fight infections. And this is probably the way neutrophils work, or the way antioxidant nutrients affect neutrophils, these general immune cells. But the other branch of the immune system is acquired, vaccination type things. And I'll get to that. I thought that was the next slide. Sorry. I'm going to run through some data now. This is all dairy cattle data. Whatever I show from this point on is dairy cattle data. It's unequivocal. Adequate selenium improves neutrophil function. Unequivocal. And I don't say that word very often. It enhances this over here. They give mastitis here on time 0, and they measure how long it takes for cells to come into the mammary gland. The faster they come in, they can kill the bacteria, and the cow returns to healthy. Selenium deficient is the red line, very, very slow response. The blue line is selenium adequate cows, rapid response, and what happens is these cows killed the E. coli. This was E. coli mastitis. Killed it very quickly. The cow never got sick. These cows, the E. coli wasn't killed, infection developed, and the cows got quite sick. So selenium enhances migration. This is kill. There's been 7, actually there's 10 studies now. 10 studies with measuring kill. This is neutrophil kill. 3/4 of the studies show a positive effect. And this negative one is an in vitro study, which probably I shouldn't even put up there. And once in a while it doesn't work, but almost always improves kill. Which again, if neutrophils kill, less mastitis. Copper often also is involved, and the data is pretty strong on copper. Phagocytosis is neutral. Copper, they phagocytize fine with or without copper. But this is kill again. 8 studies with dairy cattle. 3/4 of the studies, if I take a deficient animal and a sufficient animal, the sufficient animals had better immune function. And a lot of people interpret this. This means more and more copper is good. If a little bit helps, a lot will help more. And that's just the opposite way to think. Going from deficient to adequate improves kill. Going from adequate to excessive reduces kill. So data is very clear, provide adequate copper. And one thing here is, why does, copper or antioxidants, why are they so important to immune cells? Because they really are. We can have copper deficiency and not see much problems other than immune cells until they get really deficient. The thing with immune cells is these cells have very high polyunsaturates. These cells have to go from the blood to infection sites, so they have to be very flexible. They have to be able to sneak out of the bloodstream, get to infections. So they have very high polyunsaturates. Polyunsaturates are very sensitive to oxidative damage. So these cells, because they have all these polyunsaturates, free radical can attack and kill the cell or damage the cell. So that's why we often see no other effects other than immune cell function in these marginal deficiencies. Okay. Then the other type of of immunity is called specific immunity. This is vaccination types. You get J5, those types of things. Antioxidants affect that, but this is much less clear on mode of action. And it's probably actually a lot of this is right here. This oxidative balance may be involved, but it's probably a lot of these nutrients now actually we know have direct effects on gene expression. And so, like selenium has a huge effect on gene expression. So a lot of this on the benefits of these nutrients on specific immunity is much more complicated. It works, but we still don't exactly know why. And this is just an example here. Lymphocyte blastogenesis, that's a laboratory assay we use to measure acquired immunity. And it's just showing you that, again, the vast majority of studies where they measure this as an effect of selenium vitamin E on acquired immunity, the vast majority of them are positive. Selenium and vitamin E improves acquired immunity. The other, copper, that date is pretty, whether copper has any effect on acquired immunity is questionable. Zinc probably does. And I think I have a slide on that later. So now I'm going to be a little more specific. That was the general background. Again, there's almost no question that cows with inadequate selenium have high risk for mastitis. They're going to have higher cell counts, more clinical mastitis. The mastitis will last longer, and the mastitis will be more severe. And this has been shown in multiple countries, every continent except Antarctica, where these studies have been done. It's positive in cattle, sheep, goats, and buffalo. It's very, very consistent. Adequate selenium. So some of the questions, well, how much is adequate, and does form matter? With form, we're really looking at selenite versus selenium yeast. And this isn't, in most cases, I don't think form matters. This is a study we did a few years ago. Feeding yeast, selenium yeast, always increases blood levels, always. So this is the cow. We fed this during the dry period. Cow has higher blood selenium when fed the yeast. The newborn calf had higher selenium. The colostrum much, much higher with yeast. But we also did some immune cell function assays, which we think are better indicators of true function, and form didn't make any effect on kill. So whether, in this case, selenite and selenium yeast did just fine. They both stimulated kill. One wasn't better than the other. This is a study with beef. Again, selenite versus selenium yeast. Now they're measuring another immune cell called macrophage. I won't go into what this does. It's just another immune cell. In this study, the form mattered. Here's the selenium yeast is the green, much higher than the selenite in the blood. And here's this measurement called phagocytosis, macrophage phagocytosis. And the selenium yeast was significantly better. But most of the data doesn't show a big difference between form. So feed enough, form isn't that important. And this is the kind of wraps it up, and that's clinical mastitis, not some lab measurement, just clinical mastitis. The blue bars are quarters infected at time 0. So there was about 20% of the quarters infected, so pretty high prevalence. They fed selenite, selenium yeast, equal amounts of selenium, I think it was 0.4 parts per million, and nothing, no supplemental selenium. No change here because they didn't do anything. Mastitis still very high. Selenium yeast reduced mastitis significantly and substantially. Selenite reduced mastitis significantly and substantially. No effect, no difference between selenite and yeast. Both were very, very good at reducing mastitis, clinical mastitis. So if you have the ability, blood test, for most trace minerals, blood tests are meaningless, absolutely meaningless. Selenium is an exception. There's enough data out there, it responds to changes in diet. Most minerals don't unless they're really, really bad. And there's enough data to come up with where we think it should be. Whole blood, if you sample whole blood, somewhere around 0.16 micrograms per milliliter. If you're in that range, and you're having problems, it's probably not selenium. And these are all, I know you probably can't read these things, these are all different health measurements, and they all, right around here, is where they reach the low point. And you'll notice on some of these curves, this is retained placenta right here. It goes up with high blood selenium, and that's real. That's not a statistical fluke. Excess antioxidants are pro-oxidants. Don't forget that. Okay, vitamin E. This is usually positive. 5 studies, improved mammary gland health. 2 studies, no effect. But what I really want to talk about is this negative. 1 study showed, and it was a big study, had a negative effect. Supplemental E had a negative effect on mastitis. And I won't go into this. These are a lot of different protocols and all, but in general, the majority of studies say feeding adequate vitamin E reduces mastitis or cell count or infections. What started this was one of the first studies I did at Ohio State, where we went out to many, many farms and took blood samples and aseptic milk samples, looked for things like mastitis. And we were plotting this data, and this is multiple farms. There's several hundred cows in this graph. And what we see here is this is calving here at day 0. Blood levels are just staying right around 2, which we now know are a little low, but just sitting right around 2. And then about a week, 10 days before calving, it just drops like a rock. It just plummets. And it stays down a few days after calving, and then as these cows start eating, it goes back up. And this always, we look at that and say, what's going on here? About the same time, this is a review paper, that's why the year's off. But about the same time, this thing called immune suppression was starting to be talked about. My battery's dying here, so you may not be able to see my pointer much longer. But this is neutrophil function, and you just measure this in the lab. And it's steady, 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 steady. About a week before calving, it plummets. This is normal. And it stays low and then eventually comes back. And you could move those 2 graphs. They're almost perfectly superimposed on each other. And that got us thinking maybe one of the reasons we have this immunosuppression, our cows are just deficient in vitamin E at calving. So the question is, why does this happen? We didn't pull the vitamin E out. Why did we see this big drop? The biggest reason, there's 2 main reasons. Colostrum. Colostrum sucks a lot of vitamin E out of the plasma. But colostrum is a great source of vitamin E for that calf. A lot of people said, well, they just eat less. That's not it. We know that because we can force feed cows and blood levels still drop. It's involved a little bit, but not, the drop in intake is marginal effects on vitamin E. The other thing is calving or parturition is a very oxidative stress event. Huge amount of free radicals are produced, just huge. And so the vitamin E is doing its job and sucking up free radicals. So we actually have an increase in the requirement. Colostrum synthesis and breakdown of the free radicals associated with calving. And so we started looking at this pre-fresh period. Can we supplement extra vitamin E to prevent mastitis? It's also been done now with metritis. It also works for that. So we did one experiment where we fed 1,000 units, which is the current NRC requirement. Our negative control was 100 units. And our treatment was 1,000 units up to 2 weeks prepartum, and then we fed 4,000 units a day. And if we look at blood levels, this black line is this high vitamin E. We completely eliminated this drop in vitamin E with 4,000 units. We go over here and look at clinical data. This is intramammary gland infection. We don't see anything with the 1,000 units. They're still becoming infected, but the 1,000 units reduces mastitis, so the bacteria, or the cow can kill the bacteria. But look at, with the very high vitamin E, we significantly reduced infections, and we really reduced mastitis, clinical mastitis. Most of this is environmental streps. So a huge reduction in clinical mastitis the first 2 weeks of calving, after calving. Okay. So we think pre-fresh needs more vitamin E. And we published that paper, and then people start thinking, okay, this is, there's been 4 other studies that have done these things, and they've all been positive. But then the question was, why don't we, instead of feeding just 1,000 units for the dry period, why don't we just feed more the entire dry period? And so this group out of Belgium, and I think there was a UK investigator on this as well, set up a very, very big experiment. 5 farms. Each farm would have had 2 pens. One pen dry cows got 130 units a day, which is quite low. The other pen got 3,000, which is 3 times NRC. So they fed this during the whole dry period, not pre-fresh. And then they measured what they called clinical quarters per total quarters. And there is some issues here on how they measured things, but I don't want to get into that. But what you can see in almost every farm, these 4, farm A, B, C, and D, the high vitamin E actually had more mastitis, not less, more. The cows here in farm E read the book and knew what they were supposed to do. But this is overall 5 farms. Cows fed high vitamin E were 1.7 times more likely to get mastitis, not less, more. And again, there's some technical issues here, so I don't know if this is actually negative, but it's clearly showing no benefit. So there's absolutely no reason to feed high vitamin E the whole dry period. Don't do that. I think the data still is very good for feeding high vitamin E the last week or 2 of parturition. So why would high vitamin E cause a problem? Well, as I've been mentioning, antioxidants in excess are pro-oxidants. Vitamin E is not the most reactive chemical, but it could happen. There's data with humans, and this was with children, where again, they fed massive amounts of vitamin E. I can't remember how much, but relative to our requirement, it was massive enough. And what this did was I said earlier on, you have to, for the immune system to function, you have to produce free radicals. You have to. But in this study, what they found, they fed so many antioxidants, the concentration of free radicals never got high enough. So it could be that. The most likely thing, though, is fat-soluble vitamins. There's only many. Vitamin A and vitamin E go through the same mechanism to get absorbed. 3,000 units of vitamin E is about 500 times more molecules than the normal vitamin A. So if you feed vitamin A at approximate requirement, 3,000 units of E, that's several hundred times more molecules of vitamin E. And I just simply think there wasn't enough absorptive sites. So what they might actually have been seeing is a vitamin A deficiency caused by excess supplementation of E. So again, just stay within the norms here. Okay, copper. Again, this is the study. 7, no supplemental, 27 total. So 20 supplemental ppms. And again, the cattle fed the deficient diet when they gave cows mastitis, the infection became more established, cows got sicker. Cows that were fed adequate copper, the infection didn't get established so well. This is the cell count. Too many people interpreted this to mean cows need 27 ppms copper. No, the way to interpret this is don't feed deficient diets. There's a long way between 7 and 27. And later, I think tomorrow, I'm going to give you the right number. It's 15. 27 is too much. But this clearly says copper deficiencies increase risk of mastitis. Chromium. Is chromium legal here? No. Okay. In this study, it was 20 supplemental plus the 7 basal. And that's too much. And chromium we'll skip. Zinc methionine, a lot of studies. This is likely not due to PMN function, neutrophil function. This is either acquired immunity Or it could also be this, if you heard Ian Lean this morning talk about the microbiome. Trace minerals have effects on that which affect immunity, but this is almost certainly not an antioxidant effect. But a huge number of studies looking at the blue bars here are not zinc methionine, the yellow bars are with zinc methionine, significant reduction in somatic cell count. But again, this is likely not antioxidant. It's some other mode of action. Okay, so I want to wrap up here with this immunosuppression, using good nutrition to reduce immunosuppression. And again, I'm going to go, this will be quicker because you'll get this in other talks. NEFAs, non-esterified fatty acids, have direct negative effects on immune function. This isn't like you can't say, well, it's because they're energy deficient. No, these are direct negative effects. Ketones, BHBA, have direct negative effects on immune cells. And again, the negative energy balance helps or hurts, but these are chemicals that have direct effects. These aren't indirect. Hypocalcemia, immune cells need a lot of calcium. Under hypocalcemic conditions, these cells don't function as well. So one way you reduce mastitis, you prevent hypocalcemia, you prevent ketosis, and you prevent massive loss of body weight. Just good nutrition, and that will help reduce mastitis. And this just shows the data here. These are lymphocytes, immune cell. They incubated them in NEFAs. This is a measure of function. I won't go into that. High numbers are good. This is where we think the cutoff is for NEFAs, around 500. When it was less than that, very high numbers, very good function. When it got much lower than that, depressed function. This is fresh cows. Now this was in vitro. This is where they took blood from fresh cows and looked at, they measured the NEFA in the blood, took the cells and looked at the assay, looked at the function. So this is in vivo and a nice negative relationship. High NEFAs, in this case, this assay, low numbers are bad. So high NEFAs, reduced neutrophil function, direct effects. Ketones. And again, this is an in vitro. They take these cells from cows, incubate them in media with different levels of BHBAs. The cutoff where we get excited is right here. Somewhere between it's usually about 1.2, most people say that's an indicator of subclinical ketosis. And you can see actually when we look at immune function, somewhere between 0.1 and 1, we start seeing significant reductions in function. And again, this is just a lab measurement. High numbers are good. But it really tanks here. So when it gets higher than 1, which is subclinical, we see suppressed neutrophil function. So again, it's direct effects, not necessarily just because the cows don't have energy. And then lastly here on calcium. They know this is with lymphocytes, another form of immune cell, more acquired. One of the signals to produce antibodies is calcium. So when a lymphocyte has to produce antibodies, they release a lot of calcium. That starts a whole cascade. And it's known that if you take lymphocytes from cows that are hypocalcemic, this doesn't happen very well. They simply don't have the calcium to do what they need to do. So again, milk fever, preventing hypocalcemia, helps reduce mastitis. So to wrap up, for trace minerals and vitamins, NRC is where to start. There's some problems with NRC, but that's where you should start. And you have to have good reasons to deviate from that. There's reasons too, but you need some reasons. Okay, so general recommendations. Copper, 15 ppm total copper. That's the basal plus supplemental. In most cases is fine. But copper is one you can't just use a book. You can't just say I'm going to feed 15. And I'll cover this more tomorrow. There's a lot of antagonists in the world. And this is the one mineral where I want a lot of information before I'm going to say I'm going to feed this much. I'll cover that more tomorrow. But if there's a lot of antagonists, I might be at 20 or 25. But 15 is under normal conditions. Zinc somewhere in 50 to 80 total. Count what's in the feed plus supplemental. And again, this organic, I'm going to talk a little bit about this tomorrow as well. The effect of organic trace minerals may be different than what we always thought. We just thought it was more available. I really don't think that's it. I think it's levels it's having effects at the gut. So these might have different effects than zinc sulfate. But 50 to 80 total. Selenium, I don't know, is it regulated here? Can you feed 0.5? You should feed 0.5. We're regulated at 0.3. Is that 0.5 total diet or, okay. So feed 0.5. If I could say this in the US, I'd say 0.5. I can't say that though. So 0.5 supplemental or 0.5 selenium. These are the levels we use if we measure whole blood. I said if you measure plasma around that level. So again, if you're having a lot of mastitis, a lot of metritis, take some blood. If these are in these ranges, look elsewhere. It's not selenium. Chromium we won't go into. Vitamin E. During the dry period, 1,000 units if you're in confinement. If you're on pasture, I don't think you need any. If there's plenty of good lush pasture. 500 supplemental during the lactation period. In transition, though, I think there's very good data saying this isn't enough. The problem is there's been 4 or 5 studies, and they all use a different amount. And there's been studies where they use 2,000, 3,000, 4,000. They're all positive. Where should you be? I don't know. I normally, because vitamin E has some cost, I say normally if you're having issues with metritis, mastitis, start here. Don't start here, start here, see what happens. And again, this is confinement cattle. Pasture probably still needs some at transition, but not nearly that much. The other thing I didn't mention about is there is natural vitamin E. It's better than what the label says, at least in the US, which is regulated as what the label can say. It's much better than that. It's twice. The label says it's 1.4. Economically, it's still probably not justified. But if you get natural vitamin E, it's better than what the label says. And then lastly, don't forget ketosis, hypocalcemia, and mobilization of fat, independent ketosis, increases mastitis. So feed trace minerals and vitamins correctly, but make sure energy is good and make sure these cows eat and make sure you prevent hypocalcemia. And with that, thank you, and I'll be happy to try and answer questions. Okay.
Thank you, Bill. Very interesting indeed. I know there's going to be more tomorrow, but I'm certain that there will be some questions provoked by that. So we've got some roving mics, and if I could ask for hands up for questions. What I would do is ask you just for context to say, introduce yourself, your name, and where you're from, and it'll help us to add a context to the question. So who's going to kick off with the first question? First question in the front row. I can offer the microphone.
Thank you so much. Michael Bain from Zimpro. Just on the selenium, Bill, do you feel we should be looking at total selenium in the diet or selenium methionine levels, dependent on source?
It's all these, again, in the US, the supplemental is regulated, the basal is not. The problem, I look at total selenium. And the problem with selenomethionine is that is not the active form of selenium. And actually, I make the argument that that is a biologically bad form of methionine because it's used when you or a cow eats selenomethionine, it thinks it's methionine. That's what it thinks it is. It can't tell the difference. So if you need to make milk protein and it needs a methionine molecule and there's a selenomethionine flies by, it gets incorporated. So you see a lot of general labeling of selenium into proteins, which are not selenoproteins. And so actually, for humans, drinking milk from cows fed selenium yeast is very good. We get more selenium intake, but that doesn't help the cow. So and that's why, again, I'll go into this tomorrow, but I don't like all selenium yeast. I want a mix, half and half or 2/3 something, because to be incorporated into a selenol protein, it has to get broken down to inorganic selenium. That's absolutely essential, and then it gets built back up. Yeah. And that, in the U.S., we talked about doing that as they were more concerned with too much selenium in the human diet. That's the only reason they talked about this. And we convinced FDA that we didn't want to do that because, again, there's a state in South Dakota in the central U.S. has very, very high selenium soils. And that they couldn't sell milk if because the milk was that high. So they abandoned that. But that's a human health issue. But we also need selenium, so you make the argument, is it bad for us or good for us? But
Thank you, Bill. There's a question here in the front.
Hi, Diana Allen, nutritionist in the Southwest. Is there, in the UK, most heifers and cows are grouped together as a transition group. Are we all right in feeding the same amount of vitamin E to cows and heifers? Because I think I've read something recently where there was a big production drop with heifers.
There was. I know which paper you're talking about. That's a little confusing. It's not as clear cut as that. But again, if you're housing them together, I would not go to the high 4,000 units. I would not do that. But I don't think it's, again, on production data, I can't figure out why vitamin E, unless you increase disease, why vitamin E would reduce production. That just doesn't make sense to me. So I would not necessarily separate heifers just for that, but I would, because of that study, I would go to the 2,000, not the 4,000. It needs to be repeated because I cannot figure out the mode of action. I just cannot. And if you look more into it, it was heifers under heat stress lost. Heifers under normal condition didn't. So is this real, or when you start getting that specific, is it a real effect or something? I just don't know. But
Thank you. I think there's a question over there. Just an introduction, if I could.
Yes, Andrew Pine from Premier Nutrition. Bill, there's two questions really, if possible. One is, does stress, animal stress, reduce the functionality of the antioxidant system, or does it increase the requirement? And the second one, on the paper where the high vitamin E study increased mastitis, was the selenium status of those animals excessive, or was it deficient?
the first one is, we don't know. It increases the need for antioxidants. Stress increases the need, heat stress especially. There's good data on that, whether it's because of increased requirement or whatever, but there's good data with sheep and now with cattle under heat stress, especially with selenium, it goes up. You need more selenium with heat-stressed animals. The second one, that wasn't clear. In that paper, it wasn't clear. My guess is it was probably close to requirement, would be my guess, but it wasn't clear in that. But these were commercial dairy farms, so in my experience, they were probably pretty close.
Thank you. We've probably got time for just one more question, if there is one. The hand shot up here in the front row.
Yeah, my name is Joe Magadi from UFAC, UK. You mentioned a bit about the polyunsaturated fatty acids, and given their role in the integrity of the cell membrane and susceptibility to oxidation, is it worthwhile supplementing, say, animals during transition period with a little bit of polyunsaturated fatty acids?
There's data suggesting that there's fat supplements in the U.S. and probably here that are protected unsaturated fatty acids, and there's data showing those are positive, have positive effects. So it might be valid on feeding these. And I can never remember which one, but there's certain long-chain polyunsaturated fatty acids that have been shown to improve reproduction and reduce disease. And most of the studies, there's only been a few on it, so I shouldn't be saying most, but the studies that are generally positive are with transition animals. So it would be considered, or should be considered.
Bill, thank you very much. There is more to follow tomorrow, but I'm sure everyone would like to show their appreciation for such a great, informative talk.